Okay, I think we can start. So, good morning, everyone. It's my pleasure to start this last day of the Pangenome Biohacking 2025 workshop. And it's my pleasure to introduce you Simone Rubinacci. He is nowadays a researcher at the Institute for Molecular Medicine in Finland. He obtained his PhD at the University of Oxford. Then he moved to the University of Lausanne in Switzerland to continue his research on under the supervision of Olivier Delano in the Laboratory of System Population Genetics Group. And then he moved also to the Broad Institute of MIT. His main interest in research are about genotype imputation, haplotype phasing, population genetics. And okay, I think if you are ready, Simone, you can start. The stage is yours.
Okay, thank you very much. Good morning, everyone, and thanks, Davide and the organizers for the invitation to speak. I'm going to talk today about how identity by descent methods can help to give us insights into disease mechanisms in genome-wide association studies. So in this talk, we refer to data across hundreds of thousands of people collected in nationwide cohorts called biobanks. And this is maybe slightly different to the focus of the talks we've been seeing in the last couple of days. The most famous of such examples is the UK Biobank, a cohort with 500,000 individuals, where we have genotype data, but even more importantly, large collections of well-curated phenotypes. And the overall statistical framework to find variants that are associated to diseases is to perform genome-wide association studies, or GWAS. And the intuition behind GWAS is fairly simple. Let's assume we are interested in a disease and we want to test if a mutation is correlated with disease status. Then we can partition our cases and controls and count how many times the mutations appears in these two cohorts. And this is the reasoning behind the hypothesis testing of GWAS, leading eventually to a p-value per single variant. So GWAS have become a key tool for identifying genetic influences of human traits. And over the past 2 decades, larger GWASs have identified more and more genetic variants that associate with phenotypes. For example, GWAS of height have grown exponentially in both sample sizes and discovered associations, with the most recent GWAS analyzing over 5 million individuals and discovering more than 120,000 independent loci associated with height. So yeah, despite all these discoveries, GWAS has by no means solved genetics, but we have learned a few lessons along the way. So first, we now have hundreds of thousands of variant trait associations available in the GWAS catalog that we can query. And this is a visualization of all of them across all chromosomes. We know a lot about gene programs, and there has been a multitude of publications performing GWAS. And these associations are key for risk prediction and screening tools, and they are slowly but steadily starting to be present into the clinic as well. Also, GWAS hits have a much larger likelihood to be a successful drug targets. And so several pharmaceutical companies are interested in GWAS hits for specific diseases, as they can be then focused and carried over. There is approximately a 2 or 3 times higher likelihood to be a successful drug targets than variants obtained with other methods. Another thing we have learned is that complex traits are just complex. And that's, so for many human traits, larger GWASs have pointed us to more genetic variants that associate with tinier phenotypic differences. And unfortunately, these associations are harder to individually interpret, and this is how just polygenic traits behave. And finally, we learned that there is still a lot to learn. For example, we have only a small representation of the whole spectrum of human diversity, and we miss key classes of variants. And I will come back to this in the second part of the talk. Also, as our focus is on biobank data, this focus of this talk is on cost-effective genotyping technologies. We are all familiar with high coverage whole genome sequencing that provides a view of an individual genome by sequencing the entire DNA at high depth several times, for example, at 30x. In contrast, SNP arrays is an alternative technology that has been widely used in major biobanks, especially in the last decade, and focuses on genotyping specific non-genetic variants across the genome, around 1 million common variants. And these both are cost-effective technology for GWAS analysis. However, both short reads and SNP array data do not have access to the two, do not give access directly to the two haplotypes, the paternal and maternal component of an individual that is deployed. So we use statistical phasing techniques to actually phase the data, so to obtain the two haplotypes. And this is the process of using correlations between individuals to give us these two haplotypes. And it might seem simple, but it's a very difficult problem. First, because each heterozygous site doubles the state space, and so it quickly becomes extremely large. And on the other hand, we need very large cohorts to perform this accurately. And so, of course, this makes also things even harder. So I've been involved, as David mentioned before, to a large amount of phased release of the Biobank and different data sets, SNP array, whole exome sequencing, and whole genome sequencing. And here I will focus on whole genome sequencing release that has been finalized recently. And we do have accurate method to phase SNP array data. These are the previous generation of methods, but if we will apply these methods to phase whole genome sequencing data, this will be just not feasible. So because moving to whole genome sequencing, we have approximately 1,000-fold increase in the amount of data, which means that we phase SNP array data for 500,000 individuals in one day. This will take more than 3 years for to phase the sequencing data. So for these reasons, we had to develop new tools to phase whole genome sequencing data, and we call it ShapeIt5 as it extends the tool for SNP array data called ShapeIt4. And the idea behind ShapeIt5 is fairly simple. First, we phase common variants using a standard approach, essentially the ShapeIt4 approach, something that, as I mentioned, works very well and is very well established. So to do that, we build a haplotype probability model, and we initialize it with a random phasing at heterozygous sites. And then we find the pair of haplotype that best describe, that best can be derived from that probability model. And this is just an initialization, and it's just enough to kick in the model. And as we keep iterating and updating this probability model, we get kind of very soon to convergence, and this will be the final phasing we will report. So this is the standard way to phase common variants using statistical approaches, and after we phase these common variants, we use this as a backbone to phase rare variants on top of it. We call these common variants as a scaffold to phase rare variants. And to do that, we take each sample in turn, and we only consider rare heterozygous sites, those that we need phasing. And for a specific heterozygous variant in one individual, we select a set of other individuals in the data set that share long haplotype tracks, a long haplotype track that is identity by descent using the common variants that have already been phased. And we might need to force homozygosity sometimes, even if the individual is heterozygous, just because the data has not been phased yet. And essentially, we use probabilistic models that are very similar to the one I briefly described before to work up the probability that each of the two target haplotypes carry this rare allele. And we assign the rare allele to the most likely of these paths, here shown in green. And importantly, as we work with probabilities, we can also report confidence scores, and this can be very useful for downstream analysis. As we can see later, phasing their variance is very difficult and is more prone to errors. So it's a good way maybe to filter out variance using these confidence scores. So now that I briefly mentioned how the method works, I'm gonna show you how well this performs. And this is how the method performs for the whole genome sequencing data of the UK Biobank samples. So on the y-axis, we have a measure of phasing errors called switch error rate. So we measure this switch error rate as a function of minor allele count on the x-axis, as we want to see how the method performs at rare variances, where we know that the phasing won't work as well. But actually, given the very large sample size and the high quality of the data, you can see that the method works extremely well, even for ultra-rare variants. And just to give you an example to try to understand what I try to mean with this plot, if we get the phase right in 95% of the cases with a variant with a minor allele count above 6, which corresponds to a minor allele, a copy of the minor allele every 50,000 individuals. So we are extremely accurate even at this extremely rare variant spectrum. So we are getting to a point where phasing errors and genotyping errors are start to be indistinguishable with these very large population-based efforts. So at the end, we generated a final cohort containing 200,000 individuals and around 700 million variants, all phased. So now that we have this very large cohort, it would be interesting to know what can we do with this. And there are a very large amount of things we can look at. For example, we can look in downstream application at compound heterozygous, so loss of function on the same gene. But I'm not going to focus on that side for today. I'm going to talk how we can use this resource to actually improve other resources. So to do that, genotype imputation is probably the most common scenario, where genotype imputation is a technique that is used to infer ungenotyped variants, specifically in SNP array data, by leveraging essentially haplotype sharing into larger whole genome sequencing cohorts, such as the UK Biobank I've been showing before. So by using this as a reference panel, we can predict genotype we don't see in our data. So the most known methods to perform genotype imputation are all based on this Lee and Stevenson and Markov model and share a lot of similarities to the model I've just kind of briefly described for phasing. So these SNP array imputation methods are currently extremely efficient. They cost much less than 1 cent per genome, and they can scale on the largest whole genome sequencing cohorts that are available. And indeed, imputation accuracy depends on the size of these reference panels. The larger the reference panel, the more accurate the method is. These methods are currently sublinear in the size of the reference panel, or at least sublinear in the parts that perform heavy computation. And this is, for example, what we do with the impute5 method, where we do a linear pass using a very efficient data structure called positional Born-Soluble transform, but that after this very quick linear pass, we all have all the probability computations completely sublinear. And we recently used this impute5 method to impute the entire UK Biobank, again using Genomics England as a reference panel. Genomics England is a cohort of around 70,000 samples whole genome sequenced coming from the UK. So this is a very important resource for GWAS analysis for ultra-rare variants. And the data is being returned and is available on the UK Biobank Research Analysis platform. And interestingly, and maybe surprising to some of you, similar approaches can be used for sequencing data as well. And here I'm showing a cartoon, kind of trying to give an idea of what high coverage whole genome sequencing will look like, for example, in IGV plot, a simplified IGV plot, where a variant calling here will be fairly easy because we have high coverage individuals, especially for talking about SNP and indels in this case. However, a more cost-effective technology that has been proposed in the context of GWAS would be to decrease the coverage of this short-read sequencing data, and proposing low coverage whole genome sequencing as an alternative. For example, using 1x data, where we only have one read on average in each position of the genome. So this is what I'm trying to show in this cartoon. And this has been proposed as an alternative for SNP arrays in genome-wide association studies, but also this comes naturally in some specific application of genetics, for example, ancient DNA or circulating DNA, where the amount of DNA that is available is unfortunately very low, and we cannot do much better than 1x coverage. So with 1x coverage data, standard genotype calling is basically unfeasible, and there is a large uncertainty in the data. So how can we perform genotyping with data of this type? Well, first of all, we could model this uncertainty in a probabilistic framework using genotype likelihoods, representing the probability of the data of the reads given the genotypes. And these probabilities will be completely flat if we don't have any observed data, for example, when we have a coverage of 0, but they will be spiked towards one or the other genotype as soon as we have reads available. So the key point of this is that at a single position, we don't have much information. We have just a probabilistic representation that is maybe not particularly significant. But overall, we have enough to capture the underlying haplotypes. And therefore, using an external cohort of well-phased, high-coverage whole genome sequencing data, like the KBioBank resource I showed you before, we could use this external cohort as a reference panel and use this probabilistic assignment, these genotype likelihoods, to identify haplotypes and then perform basically standard imputation, similarly to what I've been kind of trying to give you an idea before. And by running this procedure and refining this across iterations, we could get very accurate estimates of the genotypes. And a few years ago, we proposed a method called GLIMS that solved this big computational problem with low coverage whole genome sequencing, and we made this available for the first time to very large reference panels. And essentially, the bottleneck there was that there was not there was a quadratic sampling algorithm, and for the first time, we were able to propose a linear sampling algorithm for the phasing. And we combine this with efficient data structure, such as the positional Borzoi transform. And recently, we have released a version 2 of the method, where we are also introducing sparsity in the model and specific modeling for rare variants. So we have been carefully looking at the impact of low coverage whole genome sequencing to downstream association analysis and its power to association testing. And here we show that it is more powerful than SNP arrays for coverage of around 0.5x or above in every case. And even at coverage such as 0.1x, we have a very, very good power anyway. We get a power that is more than 90% that what we will obtain with high coverage whole genome sequencing. And so what I'm showing here on the left side is some local zooms that you can see the difference between 1x and different other low coverage sequencing, but also SNP array data. And what I'm showing on the right side is just an agglomerate form, just to understand what is the power overall. So we've been looking this in the context of GWAS, as I'm showing you here, but also in the context of eQTL mapping using gene expression data, and we see a similar pattern. And finally, we also look at the power of association testing for rare burden test, which looks at rare variants specifically. And even in this case, we even show better performances. Also, low coverage whole genome sequencing is better for underrepresented populations. And this is because SNP array data has to be ascertained specifically for specific populations, while sequencing is unbiased compared to the population. So this has had quite a big impact, and these results have inspired establishment of new technologies, such as blended genome exome at the Broad Institute, concentrating sequencing efforts at exome sequencing data, having high coverage in those regions because we are interested in rare variants and rare CNVs, and offering low coverage whole genome sequencing in the remaining part of the genome, but still using the same assay, the same sequencing assay, for reducing the cost dramatically. And currently, hundreds of thousands of individuals are processed using these technologies, either in research, but also in pharmaceutical companies. They use similar assays where they genotype exome data and then use low coverage outside. So this is very likely how the future of low coverage whole genome sequencing will likely be shaped in large scale analysis. And it's very excited that method-wise, they're using the methods we have been proposing. So in the second part of the talk, I'm gonna talk a little bit how similar imputation techniques and phasing techniques can be used to improve structural variant calling in large cohorts. And this goes back to actually my introduction, where I raised a last point where I said that we miss a category of variants in GWAS. And in particular, we miss structural variants, large genetic rearrangements that affect more than 50 base pairs of DNA. And they come in many forms. They are deletions, duplications, insertions. And because these structural variants are large, they can even span several kilobases. They have a large effect on phenotypes. And indeed, many structural variants have been shown to be drivers of very important Mendelian disorders. But despite this, structural variants have been rarely included in GWAS. And this means that the impact of structural variants to more like complex traits and diseases is still largely unknown. Structural variants could reveal new associations, but also be the true cause behind a cryptic SNP association, where the SNP association is an LD with the structural variant. And the reason why this has been, the reason why structural variant have not been consistently included in GWAS is because structural variant calling is technically difficult to do at scale. Roughly only 10,000 structural variants per individual can be detected from short-read sequencing data. And recently, long-read sequencing effort and a Pangenome have provided a comprehensive look of structural variation, but just for a few hundred individuals, and not in a context of GWAS. So currently, there are several data generation efforts, both in the long-read and short-read space, to include structural variants into genome by association analysis. And here we show that actually it is possible to perform improved structural variant calling with the data we currently have, so short-read with genome sequencing and high coverage, by accurately modeling haplotype sharing in biobanks. And to be more precise, we build on previous work, and previous work has shown that from large cohorts using haplotype-informed methods, it is possible to discover and genotype specific classes of structural variants, specifically VNTRs, where we have tandem repeats, or CNVs, and uncover their effect on phenotypes. And in this work, we've been working on extending such approaches, first to detect all classes of structural variants, including insertions that are typically very difficult in short-read sequencing data, and inversions and mobile elements. And second, for scalability, extending this approach to the entire cohort of 500,000 whole genome sequencing sample in the biobank. And going a bit more into the details, the way we can detect structural variants from short-read sequencing data is to look at specific patterns in the read, such as split reads, discordant reads, or read depth patterns. And we refer these as structural variant signals in the following. And the key idea is that once we have these structural variant signals across all individuals in the cohort, we can use relatively standard phasing techniques to include structural variant signals and denoise sequencing reads, effectively allowing structural variant calling at the haplotype level. So we developed a method for this, it's called SUVI, and to perform such analysis in the UK Biobank. And in short, the method is three main components. First, we detect structural variants, then we extract these structural variant signals across all the ground files, and then finally we perform haplotype code. We perform coding of structural variants using haplotype phasing. I'm not going to go into the details on how we perform detection to the sake of time, but the key idea is that we can use the structural variants that are known, but also we can perform structural variant discovery by just binning the genome and looking at the structural variant signals. And we have a way by looking at siblings, a correlation of IBD2 segments, so segments where siblings share, they are monozygotic twins in this region. We could look if there is inheritance. If these patterns, if these signals are shared, then it will show that this is inherited. So once we detect the structural variants, then we need to extract the structural variant signals across the entire cohort. And this means processing 500,000 high coverage whole genome sequencing compiles. It means processing around 10 petabytes of data. So this, given the size of the data, this is of course the most computational intensive part of the analysis, but we heavily optimize this step and we are able to process the entire cohort for less than £1,000 in the Biobank search analysis platform. And finally, once we get these signals, we can phase them and denoise them using information across haplotypes. And at the end, we have this variant level cohort that can be directly taken to association testing. And the UK Biobank has a very large phenotypic cohort, so we can really test several phenotypes. So in our analysis, we also included European individuals from 1,000 genomes project to compare our structural variant calls with the available short reads and long read sequencing data. So we use structural variants derived from long reads that are available for a subset of individuals, and we checked how well short read methods, our SUVI approach, but also what is available from 1,000 genome using short reads, have been able to detect these high quality structural variants. And we found that our approach detected more high quality structural variants than it was there with 1,000 genomes. So after assessing this, we wanted to do association testing, and we did association testing using 57 quantitative traits in the UK Biobank. And our goal is to identify structural variant associations that are not explained by linkage disequilibrium by any nearby SNP indels. And we recently completed this association analysis. We're still finalizing some file mapping. And in the following, I'm showing a couple of examples just to give you an idea of the structural variance association we find, how they look like, and what we can do with them. So first, we identified a 2.3 kb deletion on chromosome 3 linked to liver biomarkers. So this deletion is also present in ROMAD-SV, and it's similar allele frequency in Europeans to what we detect in the UK Biobank. This deletion is strongly associated with 4 liver biomarkers, having the highest association with alanine aminotransferase, and having an effect that is 1.4 times stronger than any SNP or indels nearby. So we tested the variant association for liver disease, and we indeed verified that there is a borderline association for liver disease as well. So we believe that this variant is a strong candidate for follow-up and replication analysis in our cohort, and we are currently carrying this in all of us. Another interesting locus we identified is a VNTR in the second intron before GLT1 on chromosome 9, and it is strongly associated with serum levels, a biomarker of diseases like gout. And its association is nearly twice as strong as any nearby SNP indels. So this VNTR has a 39 base pair repeat unit and is extremely variable across individuals, with common alleles having 1 to 6 copies. And these 6 alleles are also visually present in high-resolution assemblies from the Human Pangenome Reference Consortium. So we observe an allelic series for this VNTR, where longer VNTR alleles are linked to higher urate levels and greater gout risk. And the best target SNP for this is a topic UTL for before GLT1, suggesting that the VNTR might affect gene expression. So these findings kind of motivates us to study, kind of use GTEx to find map this eQTL signal and explore whether this VNTR can influence splicing. So to summarize, the methods I've shown you today provide an overview on how shared patterns between individuals in large-scale biobanks can help informing us of disease status, disease genetics. However, we are far from done. We still have, we only explain a small fraction of these trait associations, and there are many directions that remain for further work. As many speakers have been mentioning in the last couple of days, we won't be able to completely assess the effect of structural variants using structural variants until we have long-read sequencing data. And some of the algorithms that have been presented in the last couple of days will definitely help to reach this goal. And we need more data and better data. This also means that it's a great time to be a statistical genetics, and we will have a lot to discover in the coming years. And in Finland, we have a fantastic data set, which is FinGen, with many potential new discoveries in the years to come. And this concludes my talk. I just would like to mention that I'm currently looking for PhDs or postdocs to join my lab. So if you're interested in this type lines of research, please feel free to contact me. But also, our institute also a fantastic PhD program that allows rotation between several labs, and it's a great opportunity for students. And the deadline is just in 2 weeks. So go and check it out if interested. Thank you very much for listening. I'm very happy to take any questions.
Thank you so much, Simone. For the audience, I remember you that you can post your questions on the Matrix chat. And for Simone, we have about 20-30 seconds of delay between our meeting room and the live stream. So maybe you can wait. In the meantime, I've just 2 questions. Both of them are probably very silly, but the first one, regarding the low coverage approach, probably I missed the point. Do you have a sort of threshold, a lower bounds under you are not able to perform very well, or?
Yeah, that's definitely true. That threshold will be probably around 0.1x. I think under that, when we tested, things will start to become very difficult. So it's still usable to some extent, low coverage with genome sequencing data less than 0.1x, but it will require probably even larger reference panels, or maybe imputation is not the best approach because we start not being able to capture haplotypes anymore, at least with the current methods we have now. But until 1x, especially if we have a good reference panel, we can do extremely well, and that was maybe surprising, and I wasn't even expecting it myself when I started working on this.
It's very surprising. I agree with you. The second question is just out of curiosity and more in the context of this workshop. Are you aware of any approach based on Pangenome graph, or I don't know, GWAS or some topics related?
That's a good question. I'm sure that there are people thinking about this. So now maybe more in the context, more than GWAS, more like how to use the Pangenome graph to perform imputation. And I think that's definitely there are some methods that perform this in the context of maybe not Pangenome directly, but personalized reference, essentially. So the idea is to perform imputation and then create your own personalized reference, but now you have the alternative alleles in your personalized reference, and then remap your reads essentially into this personalized reference. So definitely there is I'm confident that there are many people thinking about this very carefully, and potentially also in the context of pangenomes. But I'm not sure about connecting this directly to GWAS, because, well, of course, when the pangenome will give us access to darker regions of the genome. And so that's definitely something people will want to do to understand what is the impact of this structural variants and this dark region of the genomes in terms of phenotypic effects.
Okay. Thank you again. I don't see any question on Matt's chat. Let me check another time. I don't have any question. If I miss some question, you can just post them later and we will forward to Simona. So thank you so much again. Now we will have a 25 minutes break, more or less, because next talk we start at 10 Italian time. So thank you again and see you.
Thank you. Bye.
Hi. Ciao.
Okay. Can you hear me now?
Yes, I can.
Okay. Hi. Hi, Anna Paula. So
Hi.
we are, you can try to present
Yeah.
in,
Yeah, I'll share my screen in a sec. Okay. I do share. Yeah. Okay. I don't know what that means, but sorry, I just need to change the settings because it doesn't let me share my screen with Safari. 1 second.
take your time, no problem.
I actually never use Google Meet. That's why. Hold on. How do I change it? Settings. Oh, maybe here. Actually. Settings. Turn it on. 1 second. So does everybody can hear already, or because I only see you guys, but there are like people live.
Yes, no, we are going to stream live on YouTube, and so the audience will be on YouTube while we are here just to take care of the presentation and of technical issues. Okay. Yes, but what I see is a blank screen and the mouse, the pointer. You are muted. You are still muted in case. Okay, now I see your desktop.
Okay.
Yes.
Okay.
Maybe try to start the presentation. Go to the next slide so that we check if everything is fine. That's perfect. That's perfect.
Okay,
so
thank you.
We can start. So it is my pleasure to introduce Anna Paula Carrieri to this workshop. Anna Paula has obtained her PhD in computer science from University of Milano Bicocca, and since then she has started a very successful career at IBM UK, I can say that. And so please, we are very welcome to have you here. Please go with your presentation. Thanks again.
Thank you. Thank you for the introduction. Hi, everyone. I'm Anna. I can only see the slides, so I have no idea who am I speaking with, but nice to talk to you today. So, I'm Anna, and I work for IBM Research in the UK. I've done my PhD in University of Bicocca, but then it was 10 years ago, yes, I started working in IBM Research, and now I'm a manager and a senior research scientist in research. I manage a team of scientists and researchers that work mostly on AI for healthcare life sciences. And today I will present some work that relates to pangenomes, but maybe I'll present one project that's specifically on pangenomes, but the work that we are doing in IBM research, especially around biomedical foundation models for genomics. So this is just an outline what I'm going to talk about. So we'll first introduce IBM research very quickly, because not everybody might know what we do. Then I'll talk about our healthcare life science strategy at high level, and then I'll introduce GenAI and foundation models, and then their application to drug discovery specifically, and the biomedical foundation model the IBM global team has developed, and it's now open source. And then I will talk about a specific case of how we can apply or use foundation models for omics data, a use case for transcriptomic, and at the end, I will give a brief explanation of a pangenomics project that we are doing. We are almost completely with one of our clients. Okay, so IBM research represents 1% of the entire IBM. So we're about 3,000 researchers spread across the globe, as you can see here with the blue blots. As I said, myself and my team, we are based in the UK. And but we work very closely with a global team, so the effort is global. And this is the bit where we show off typically, saying that since our first IBM research lab opened in 1945, we've published over 100,000 papers. So this is just to say, yes, we are a company, but we are a research division. So we really care about publications and delivering that research output. And some of our researchers, this from 1945, obviously, won 6 Nobel Prizes and 6 Turing Awards, and we have patented over 150,000 patents. So now this is the bit where Wishoff is finished, I promised. What we are doing now is developing technologies that combine AI, quantum, hybrid cloud, semiconductors to accelerate scientific discovery in different areas. And one of the areas, healthcare life sciences, which is what interests us today, but we're also working in climate and sustainability and computing and nanotechnology. So before I start talking about what we do, I just wanted to give a shout out to my team in the UK. We are based in Daresbury, which is near Manchester, well, in between Manchester and Liverpool, and in London as well. And the team has different skills in AI, software engineering, computational genomics, and bioinformatics. And obviously the work I'm going to talk about is my team working on this, myself, but also the global team. So our healthcare life science strategy at the moment in IBM research is all about drug discovery and precision medicine. So how we can develop advanced technology, GenAI, quantum technologies to understand diseases, for improving prevention, detection, and treatment, to stratify patients based on similar characteristics. And there is also a big emphasis on drug discovery. So we focus on early stages of drug discovery, for example, wanting to identify or discover new drug targets, or designing de novo designing of new small molecules, new biologics, such as antibodies, or for a vaccine for influenza and so on, and predicting their property. But I'll show an example later of what I'm talking about. So this is what our typical technology stack, or what actually we do. So we analyze different type of data, different type of omics. So especially in the UK, we have a big emphasis on the omics side. So it can be human genomics, transcriptomics, proteomics, but also in the past, we've done a lot of work on microbiome data. And what we do is that we analyze these data individually, or we integrate them, or try to integrate them, even with other data types, like the other modalities, such as tabular data, images, and so on. So obviously, there are a few steps of data engineering, feature engineering, integration, and then development of different type of technology. So from an application, so for example, for classical machine learning, deep learning, bioinformatics approaches, graph neural network, network-based approaches, but the emphasis is on the moment, and it's what like the healthcare life science community is looking at, is really the development of biomedical foundation models. So I will focus this presentation on that. We also started exploring quantum application for omics data with a few clients and a few projects. And the other emphasis, or the other focus that we have, it's not only applying these AI machine learning models, but also provide explainability, interpretation of the results, obviously evaluation and so on. But I will give you a concrete example. So before I go into explaining what we do around biomedical foundation model, I just wanted to share a classical machine learning, or a classical automated AI pipeline that we recently published. This is was just end of last year. So this is an open source tool. So if you want to run a classical machine learning analysis on some tabular data, they can be omics data, gene expression, they can be SNP, they can be a microbiome table, abundances of microbiomes, any tabular data. They can even be clinical data, but you can get this tool, you can run it, use some config file in a completely automated way, and you can run several classical machine learning methods. Random forest, neural network, XGBoost, support vector machine. There are a few options to do a prediction task that can be classification or regression, depending on your problem, and then you can also generate explainability results. And you can do this all, by running this tool, which is completely automated. So this was mainly, we open sourced this tool because we developed while working with many clients. And then when a year and a half ago, we moved into biomedical foundation models, we wrapped this work up and we open source it. For the community to use it, and we still use it when we want to compare the results of foundation models with traditional machine learning. But we are not developing we are maintaining it, but not developing it further, because our attention our focus is shifted on foundation model. So now, possibly many of you already know all of this, but I thought that it'd be nice to just give a brief overview of the story of AI, and now we got to foundation models. So we started with rule-based system, expert system, and then there was a big boom of traditional machine learning model. I mentioned some, random forest, support vector machine, that were able to make predictions from some data. Could have been tabular images, so on. And then there has been a lot of focus and attention on deep learning models, so deep neural network, that not only provide improved predictions compared to traditional machine learning models, assume you have enough data to train them properly, but also were able to learn from the data representation of the data that were task specific. Then in 2017, a paper was published by Google Research and DeepMind, I think, and was called "Attention is All You Need." Well, actually, it was a paper proceeding to a conference called NeurIPS, which is a very important conference in the AI community. I'm sure you all know of it. But so this paper was published by Google, where they presented a new architecture, a new neural network architecture called the transformer. This was the first transformer architecture, which is mainly at the base of, sorry, I've got a cough, which is mainly at, it's the foundation of what a foundation model is and what Gen AI is. And since then, there has been like a boom in the scientific community in developing this Gen AI method. So this transformer is at the foundation of things like ChatGPT, that is a foundation model based on language, and others, and many other large language models and foundation models. So what's different compared to this, and I will go a little bit more in detail in the next few slides, is that compared to deep learning and classical machine learning methods, not only they hopefully improve the prediction accuracy, the prediction performance, but also are able to generalize better. So learn better, more generalizable representation of the data. So let's see the difference then. So with classical machine learning or AI methods, you have a specific use case or task you want to predict. It can be a classification. I don't know, if we think about disease subtypes, it can be a regression task if you want to predict like a value and a number, degree of something. I don't know how well somebody is responding to a drug, for example. But the task is specific, and you train each machine learning model with a specific training data and to perform a specific task. So now what happened is that what happened with classical AI models that are not Gen AI is then even if you might build or you might generate a model that has a very high accuracy for your specific task, for your specific study and data set, what we found is that, and what the community has found, is that when you try to transfer that learning or applying this model on another study, in another cohort, let alone another task, the performance dramatically decreases. So these machine learning models are not able to generalize. They don't learn often, especially when the dimension of the data set is small, they often learn characteristic of the samples in your data set rather than general characteristic of the population or general characteristic of the problem that you are investigating or you want to characterize. So with foundation model instead, the idea is that you have one big model called foundation model or large language model. I mean, large language model are foundation models specific to languages. And you've got one big model and you can train it on a huge amount of unlabeled data that they could have like different modalities. And the idea is that the foundation, at a very high level, the foundation model learns characteristic of the data, general characteristic of the data, and the data can be from different domain, different cohort, different studies, just a huge amount. And then once you have this one big model, you can have different downstream tasks, and you can do something that's called domain adaptation and fine-tuning to tune these big models to your specific downstream task. And the idea is that because the model has learned generally before, the results, your prediction, are your insights are more robust and more general, and so on. So in healthcare life sciences, why this can be really it can be an advantage using these large models, because often when you deal with data, omics data, for example, genomics data or patients' genomic data. So if you think about what is one of the projects we've been working on recently, transcriptomic data of epilepsy patients taken from the brain. I mean, how many patients data can you have? We are talking about very small cohort because these data are difficult to collect. They're expensive to collect. So the clients or the pharma company or the hospital that we work with, they might have very small cohort, high quality small cohort of omics data. And if we apply a normal traditional machine learning models to this small cohort, the accuracy might be very good, but it's not an indication of the fact that we are getting robust and generalizable insights. Because it's just specific to that small cohort. So the idea is that if we have a large model, for example, foundation model trained on a very large data set, then we can fine tune it and we can leverage, we can adapt it and fine tune it for our specific task, and we can leverage small cohorts by just using this large model to do the inference on the small cohort. So in summary, the idea and the hope, and there is loads of research going on at the moment. It's not only IBM. Oh my God, I would never make that claim, but there are, every week there is a new paper coming out, often open source, mostly open source, or like foundation model for omics data or for biological data in general. And the advantage that what we are trying to see if how adaptable, versatile, these models are, thanks to this fine-tuning that you can do to the specific downstream task. So now to our IBM biomedical foundation model, which has been recently open sourced. It was like end of last year. You can see, we open sourced part of what we are developing because we are still making progress on it. And this, I want to emphasize, this is a global effort. So it's not only the UK team involved in this development, but it's a global effort in US, in Israel, in Japan, and so on. So so this is you can find our open source biomedical foundation model on Hugging Face, and I will go a little bit more into the detail of it, but you can see it, it's composed by 3 main elements, and one is target, which is all around omics data, and analyzing omics data to make prediction and find, for example, drug target. And biologics, this is to generate protein sequence and so on, or small molecules to generate new small molecules and predict their property as well. And we are working on the development of this biomedical foundation model with many partners. And here some are mentioned, but they're not all the partners we're working at the moment. So what are the advantages of as I said, there are many open source foundation models, and every week ones come out. So it's a very crowded, research space, and very competitive, but everybody is looking at this. But what is that our foundation model does? It's multimodal and multimodal. So it can handle different modalities of data. Currently 7, where we're applying to expand to 10. And multimodal because there are different, they're combined different foundation model and architecture and so on. He allows to discover new biologics, new small molecules, and new targets, where targets are like omics. You can think about targets like omics features, like a SNP or a gene with this expression and so on. And I'll give you a concrete example of what I mean by that, because I'll focus on the targets more, given that this is a Pangenomics workshop. And once we apply these foundation models to the problem of our client, and then we validate the results that we have from our foundation model. How we validate the results. And when I mean the results, I mean not only like the prediction and the accuracy, but also if we are generating a small molecule, is the small molecule can be actually made in a lab? Is it actually proper? Or how do we validate this? Well, we validate in silico, for example, with comparison with other AI model. As I said, we can always compare our results with traditional machine learning, or we can make physics-based simulation. But also, when we work with our partners, they have a wet lab, so they can actually validate in the lab our result when we generate new small molecules, new antibodies, and so on. Okay, so this is the biologics one. Very quickly, because I'm conscious of the time, and I want to focus more on the genomics one. But this is mainly, again, this is Gen AI, from a proper Gen AI model, because it generates novel proteins, antibodies, vaccine, peptides, and so on, and predict their properties. And you can see here on the top some properties that can be predicted: binding affinity, selectivity prediction, and so on. The small molecules, different architecture in a very similar way. is trained on a different representation of small molecules, 3D structures, smile strings, images, and it generates a de novo representation of new small molecules mainly that then get validated in the lab. First to virtual screening, and then the one that passed the virtual screening gets validated in the lab. But I don't want to diverge too much from the omics, or at least genomics topic that we have, and I want to move on the targets, so the biomedical foundation models for omics. So if we think about Gen AI model and natural language, we can think about a large language model in the context of omics, or a foundation model as a neural network, a complex neural network with an architecture called transformer. It's an encoder-decoder, it's a decoder-encoder architecture with a mechanism, or a mathematical mechanism called attention, that allows the model to learn the context. So it's got an attention window and allow the model to learn the context of a word, for example, if you think about natural language. And in that way, by learning the context like the human brain will do, is able to fill a gap. So predict the word that is missing. Like in this example, you can see, I made a slice of toast, but forgot the, can you grab it from me from the bridge? That's probably butter, isn't it? So based on the context, like the human brain will do, is able to predict that word that is butter. Similarly, if we think about DNA sequences, you can mask nucleotides, a few nucleotides, and train a transformer, a large language model based on DNA, to predict, to fill that gap. If you think about gene expression data, where we have genes with their expression, we can mask the expression of a gene, or we can mask the name of a gene and learn and teach and train a large language model to predict either the expression or the name of the gene that is missing. So once a large language model or foundation model that is, for example, trained on DNA data or RNA data, is trained on a huge amount of omics data, it learns characteristic of the DNA and of the RNA. And after it's done that, you can have one last layer of that big architecture, the big trained model, that can be a softmax layer, which is the layer that enables you to do a prediction. So for example, a classification. And I will give a specific use case to kind of clear this up. But I wanted to emphasize that we are developing, so part of it is open source, the single cell RNA model is open source, but what we are focusing our effort is at the moment in two transformers architecture, so two foundation models, one for genomics and one for transcriptomic, where for transcriptomic we've got both single cell RNA and bulk RNA data. The genomic model, so if you think about the prediction, what the last layer of this big complex neural network does, if you think about the genome model, imagine being able to predict disease risk of a patient based on their SNP, based on the variation in the DNA sequence. Or imagine to be able to predict the gene disease association based on the SNPs. While for the transcriptome model, we are mainly fine-tuning the model for other different tasks. One is gene perturbation prediction. The other one is drug response and also cell type annotation. Well, cell type annotation is specific to single cell transcriptomic data. Okay, so this is what we have been developing. Part of this is not open source yet, but it will be open source as well soon. And I will go forward with like a transcriptomic use case. I've got still a bit of time, and to kind of leave for the question. Okay, so to talk about our transcriptomic use case, I will first mention a previous paper that we published with one of our clients we work with called Represent. So this is a paper from 2022, I think. So it's like an old work that we did, like a previous work that we did. Here we used traditional machine learning, and in particular we used that tool called AutoXI for omics that I showed at the beginning of the presentation, like traditional machine learning, classical methods. So the problem here, we had data, patient data from our clients. These were patients affected by IBD, so inflammatory bowel disease, either Crohn or colitis. And we had genomics data, we had transcriptomic data, we had all match, obviously, and then we had patient information such as their medical history, demographics, and the medicine they were prescribed. So what we did is that we developed a method to integrate demographic, the multiomics data, and medicinal data of patients to predict, from the integration of this data, to predict drug response. So how, after the application of a specific drug to a real tissue, like a real tissue taken post-surgery, how that the patient responded to the drug. So you can think of it as a value, and you are predicting like a higher or lower response to the drug, so a better or worse response to the drug. So that was the classification task using traditional machine learning. We also applied some explainability to see of our multimodal features, which one were driven the prediction, which were most influential to drive that prediction. And we found that, for example, for patients that had colitis rather than Crohn, the response to the drug was worse. For patients that were women, the response to the drug was better. This obviously suggested by the machine learning model. And some of these insights are actually, validate the model because they are consistent with what's already known about the problem. But some of them could be new. For example, we found a few SNPs that were most influential for predicting a better or a worse response. And some of these can be known, but some of these can be novel. So that's why you kind of discover new drug target, like what's the novel candidate that a biologist or a clinician can go and look and investigate in the lab to see if that association between having a variation in your DNA and having a better response to the drug is valid and is novel. The problem with this work is that it's only, and it's not that I want to minimize it, but it is a proof of concept. And the reason it's a proof of concept is because it's a study with only like 50 patients. So obviously, whatever insight, we had decent accuracy, we had decent insights, but whatever insights we've got are not generalizable. You cannot make a big claim on a big discovery with 25 or 50 patients. So what we did is that we then moved into biomedical foundation models, and we tried to see, okay, if we use a biomedical foundation model trained on transcriptomic data, so on gene expression data, to solve the same task, does that give me better results in terms of performance, and does that give me better results in terms of discovering new features, new targets? One thing I have to mention is that this published work, this is based on genomics data, while what I'm talking now is another piece of work that is very similar, predicting drug response, but instead of from genomics data, so instead of using SNPs, we are looking at genes and their expression. But the concept is very similar. So we have a biomedical foundation model, our biomedical foundation model, trained on single cell transcriptomic data, adapted to bulk RNA data, adapted to the domain of predicting disease subtype for that particular disease, and then finally fine-tuned to predict I don't know, Crohn versus colitis, or a specific drug response, and then used for inference with our small cohort of, say, 25 or 50 patients. So, so this is an example of how you do those steps. So, for example, and how much data you need to do that. So, for the pre-training, if you're looking at, here's an example with single cell data, because the work on foundation models for this particular project is not published, I cannot like give the specific of it. When you work in a company, it works like that. You know, stuff are confidential until published, if published. But so this is a bit general, but it kind of gives you an idea of what you do and how much data you need for a foundation models. So for example, you can do the pre-training on general transcriptomic data using more than 10 million cells. Then you do the domain adaptation, where you use 100,000 cells to adapt your general foundation model on transcriptomic data to adapt it to the disease of IBD, inflammatory bowel disease. Then you do the fine-tuning with 10,000 cells to fine-tune the model to be able to predict specifically if a patient has Crohn or colitis from their transcriptomic data. And finally, once you fine-tune your model and you're happy with the performance, you can use the Earthmos data set of 25 to 50 patients and use that model to make an inference, to just predict Crohn versus colitis from from the gene expression of those patients using your fine-tuned model. And in that case, you know, to do the inference, you might need 1,000 cells, but you could also use 50 because it's inference, is downstream task. And this is just, you know, again, I'm being very vague here because we can't present via results, but this is an example in which where we are now with some of the things that we are doing, where we can see an improved inaccuracies of 2% using biomedical foundation model rather than classical machine learning. The other things that we do is looking at what genes, if we're looking at gene expression, what genes, so which features are more enriched in one disease rather than another. And you can see on the left a rank of disease that are either enriched in one disease A, which is the blue disease, or disease B, that is the orange disease. But we also look at pathways, so where these genes are more enriched in specific pathways. But while here we have ordered the gene, like we have took the top 15 gene of the rank, and we see where these genes are more enriched in a specific pathway, because that can give more biological insights and explainability to your machine learning model results. Okay, so now just to kind of connect to the Pangenomics biohacking workshop, I will talk about a Pangenomics use case. So a project that we've been working on last year that is now is concluding, but we haven't published yet. So because we haven't published, I can talk about it high level. We haven't published yet, but also I have to be honest, I have not been involved in this work. The person that is leading the work is Laura, which is both Laura and James are in my team, and they are leading the work from two different perspectives. Laura is a researcher, James is a software engineer, but Laura is currently on maternity leave. But I just wanted to give a shout out. But I can kind of explain what we are doing there at high level. So we are working with Erlam Institute, which is a big research institute in the UK, and Ragti Seeds as well. And what we are developing for them is a proper software, like software engineered software with bioinformatics capability and machine learning capabilities So you can see kind of the schematic architecture on the right here. But from a scientific point of view, the idea is to generate, what this tool can do automatically is generate a reference pangenome for an organism or multiple organisms of interest, and then align the reads of the pangenome, the reads to this pangenome reference and identify genetic variants such as SNPs. So then you can use those SNPs and apply machine learning model to predict different phenotypes from these variants, from these SNPs. And all this all gets done automatically. So you're thinking, okay, this is okay, but why are you doing it? Well, as you probably know, generating a pangenome, identify genetic variation with a pangenome or reference is a very expensive task computationally. So the idea here, this is a very much a software engineering project where we are really building a robust and fast tool to do this reference pangenomes for our client analysis. So the emphasis is on the automation of the analysis at scale, because we have many data here. And also this Erlam Institute, in particular, our collaborator, we are looking at plant genomes that, as you probably know, are way much bigger than human genomes. So generating a pangenomes for plant is more complicated than generating a pangenome for humans. And therefore, if you can do it from plants and you have enough resources and you have a software that is robust enough to do it on plants, then you can probably transfer it to a human genome as well. And then to provide our research collaborator a tool with a nice interface, a front end for non-AI or machine learning expert, so then they can be able to run this analysis independently and accelerate their scientific discovery that way. So this project is about to be completed, and the software is been now it's been evaluated by the client under an evaluation license. So hopefully a publication will come out of it as well. But you know, it's an ongoing project, so we haven't published it yet. But having said that, for the machine learning part of it, at the moment, we are using traditional machine learning, but one can think of using, again, biomedical foundation models to do the same task. So predicting phenotypes from SNP. You can use traditional machine learning, or you could potentially use biomedical foundation models. But at the moment, this project is more on the software engineering side. And we're just using traditional machine learning to start with, because it's complex enough. Okay, so this was me. And I don't know if, how does this work, if people have questions?
Yes, we are collecting the questions on the Matrix chat. There is no one so far, but there always a bit of a lag between your presentation and what the audience will see on YouTube.
Okay.
So I will start with that question. So at the beginning, you mentioned that the introduction of the transformer architecture has changed the deep learning or the machine learning field.
Yeah.
I would like to know, in your opinion, how likely it is that there will be another architecture that will change so dramatically the field?
This is an interesting question. I would say likely, because, for example, this transformer architecture at the moment is very good with languages and code. So if we think about ChatGPT or others, large language models that are open source, Llama and so on, they are very good. I don't know if you guys ever used it, but they can do loads for you. You can ask any question, you can kind of give a piece of like text and say, can you summarize it? Or maybe I shouldn't say these things. Or can you create a bullet point? Can you polish an email? Obviously, if you're doing stuff in English, it could be like quite useful. If you want to send like an email to, I don't know, a professor in English, you can like check it on Llama, or you can use IBM large language model, which is open source, which is called Granite. Please use IBM. Version 3 is open source. So IBM has, because we work in life science, we use Granite. My team is not involved in the development of Granite, but is a large language model like Llama, ChatGPT. And these models are proving to be very, very successful in what they do. They still do something called hallucination. So they're not perfect, because obviously, depending on what they're trained, they can do mistakes. So they're not perfect, but they're achieving very, very good performance. And every so often, a new version with a new billions parameter comes out, and it's like a race between Llama and Granite and ChatGPT. It's just like, it's mental. So they're really promising, but they're not perfect, one. And two, so there could always be an improvement, like in research. And two, while they're very good in languages like natural language, the language that we speak, English, in life sciences and healthcare specifically, we are still like catching up. We are still trying kind of to really demonstrate the value of this, like 2%, but not only us, but other publications. Okay, so 2% performance is increased. Is it worth it, all this effort of training, computation? Like, we are really trying to demonstrate the value in other domain that it's not natural language. So I wouldn't be surprised if, in a little while there comes up another architecture that is more suited for it. This is a variation of it, more suited for other modalities, or a better architecture that improves also the natural language foundation models.
Okay, thanks. And then I have another question. So when you say that your software is available, do you mean that the trained model is available, or that it is actually possible it is feasible in practice to train the same architecture on different data?
I'm trying to understand.
So the point is, so when you say that your software is available, do you mean that it is the pre-trained model available?
Yeah, so it's everything. So, okay, so I understand the question now, sorry. So if you go on Hugging Face, which is this, so if you go on Hugging Face, this tooling on Hugging Face, it's two models, right? You can download the weights of the model. So that's your pre-train. That's what you need to build your pre-trained model. So yes, you've got the pre-trained model. Also, at those links, you will find the link of the GitHub repository with all the code. So all the code is available. So what you can do is that you can take the pre-trained model and fine-tune it, or use it for inference, play with it, fine-tune it. You can train it further if you've got the resources. Or if you've got the resources, you can take the code and train everything from scratch on something else. But everything is available: code, weights for the pre-trained model.
So yes, part of my question was, is it actually feasible for someone else to train the model, or do we need IBM scalar resources?
No, it is feasible. You don't need IBM GPUs, but or IBM hardware to do it. You can do it in any hardware. But there is a manual that explains how to do it. A warning is that obviously the training of this, or tuning of this model, like even the inference is quite, like, because they're big models, it's quite expensive computation. Like, you need resources. So but for example, some of these models we have trained and tuned on AWS, like you don't have to do it necessarily on IBM system, no, but you'll need resources like GPUs, probably multiple.
Okay. Thank you very much. I think that it was very, very clear and very exciting. So we thank you, Anna Paula, again for having accepted our invitation. Thanks.
Thank you.
Thank you very much. And
the
session will resume in 10 minutes.
Bye-bye, everybody. Have a nice weekend. Bye.
Next slide.
Let's say something, Gianluca.
Next slide, please.
Ah, it works.
Okay. Okay. Are you ready?
Yes.
Okay. So it is my pleasure to introduce Davide Cozzi, who is a PhD student at the University of Milano Bicocca in our group. Davide will describe some of the research problems and of the recent advancement in data structures in pangenomics. As you can imagine, we have a huge problem in pangenomics, that is the data is very large and it is difficult to query and it is difficult to extract efficient information out of that data. And so Davide will show you what techniques must be used in order to access and store those data efficiently. So thanks again, Davide, for giving this talk, and please go.
Thank you, Professor Della Vedova. Good morning. I'm Davide Cozzi. As before, I'm a PhD student here at University of Milano Bicocca, and today I will talk about indexing pangenomic haplotype data using the positional Barouda transform. This will be the schedule for this talk. First of all, I will describe what is the positional Barouda transform, the PBWT. And if you remember the talk by Simone Rubinacci, it was also introduced at some point in his slides. Then we will see a sort of overview of some computational problems that are solved using this particular data structure. We will see new research topics on improvement the PBWT. We will see some extension, and more in detail, we will understand what I have done in my first 2 years of PhD, because I will describe my more PBWT. And then we will see some examples of real-world uses of this data structure. You have seen this kind of pipeline many times in these first 3 days. We can think about our input data as a set of haplotype strings, but we want just to focus exactly on the SNPs on variation site. in order to not study and not store in memory entire sequences, but just the minimum differences between them in order to have a feasible computational data to analyze. So we are computer scientists, and we need to understand what we are trying to index and store. In our case, we are looking for binary metrics, where each row represents an haplotype, and each column biallelic site. So in this first part of the presentation, we will just focus on binary metrics with an alphabet of 0 and 1, but we will see that there are some improvements in extending also the alphabet size. Then we have two main classes of problems. The first one involves find matching, find similarities between the haplotypes inside the reference panel. But we can also consider, as you see in this slide, an external query, so a query haplotype, that we want to compare it against our reference panel. On blue, I have just highlighted some of the possible matches that are interesting in analyzing this kind of binary metrics, and they are called set maximal set matches, but we will describe more in details later. So I know this will be how can I say, a more algorithmic talk instead of all the other ones in these first 3 days, but I think it should be interesting to also understand how data structure works, and maybe someone will find this kind of methods interesting and will start some new research project or include in their current research project. So we are studying haplotype as binary W-long sequences. A panel of haplotypes is just a set of these haplotype sequences. And we are interested in external haplotype that has the same length of any other haplotype in our reference panel. To give a sort of abstraction of what is a match, we can define a set match as a triple ABE, where A is a set of haplotypes, and with this triple we represent a match between these two columns, B and E. Then we can define more strictly a set maximal exact match. In this case, it's with an external query, but we can think about Z as another haplotype in our reference panel. And this kind of set match, as you can see in these 3 dots in the last block, this match cannot be extended neither to the right or to the left, and we cannot include any other haplotype without losing this property of having a match. So, positional Barrow-Wheeler transform. It was introduced by Richard Darwin in 2014 as an interesting data structure able to store and query this kind of binary metrics, and is strongly based, as you can understand by the name, on the ideas of the Barrow-Wheeler transform. Also, Richard Darwin just focused on binary metrics. How does it work? Consider that as the classical Barrow-Wheeler transform, we want to permute the input metrics in order to allow efficient queries. So it works using this colexicographical ordering. So at each column, we reorder from the right to the left. This is colexicographical, in order to make similar rows consecutive in our permutation. Let's see an example, because it's really hard to understand without an example. We are trying to index, for example, column 5. So we sort all the rows up to the previous column, in this case, the column 4, and we store the ordering of this of the haplotypes in the so-called prefix array, because the PPWT is just a set of 2 arrays per column, de facto. Then we can store the common length between 2 consecutive rows in this reordering, the common length of just the starting column, such as in the example. And as you can see, thanks to this kind of reordering, similar rows are consecutive, so they share along a common suffix. This property is also useful because if we take the column 5 permuted thanks to this reordering, obviously this is an example, but just imagine real data such as the ones from the 1000 Genome Project, where we have very few ones. We obtain that similar rows are consecutive up to a certain column, so we can likely expect that also the next column will 2 rows will share the same bit. So this produces the so-called runs, and this is the key point of how to make efficient in memory the PPWT. In this example, we can see that, for example, we have a run of 2s, then a single 1, a single 0, a run of 4 1s, and a run of a lot of 0s. We can store just the number of runs and the bit represented in each run, and obviously the length of the run. Okay, again, we are computer scientists. What does it mean, efficient? In this case, efficient means that we can compute all the set of prefix arrays, all the set of divergence arrays, and also each PPWT column. And if we consider all the set of PPWT column, we obtain the PPWT matrix in a time that is linear in the size of the matrix. And it's not something so, how can I say, easy to obtain. I mean, before 2014, it was not so simple to just produce an index in linear time that is so efficient in allowing queries. Okay, we have indexed our panel, but what we can do with this index? Richard Arpin proposed 3 main problems that can be solved. First, find all matches within the reference panel longer than a minimum length. So we are talking about all versus all haplotype matching with a threshold on the length. Then we can find all the set maximal matches, again, within the reference panel. And we will see very soon how many of these matches can we get from also a toy example. So just imagine that how efficient should be this data structure to allow this kind of solutions on real world data. And then we also can find set maximal exact matches from an external query to the reference panel. This is the entire set of set maximal matches within this toy example panel. As you can see, they are a lot. So just imagine how many they can be on, for example, 1000 Genome Project data, chromosome 1, where we have about 5,000 rows and 6 million columns. On the other hand, if the matching is guided by an external haplotype, the number of matches obviously decrease. And as you can see in this example, we have a match starting from column 0 to column 5, shared with 4 rows in our reference panel. Then we have an overlapping match with just 1 row, another overlapping match with just 1 row, and finally other 3 rows that share a match with the query. As said before, we say efficient, but how much these algorithms are efficient? For the L-long matches, the Richard Darwin proposed an algorithm that runs on, okay, this is the real complexity, but just focus on the HW sides. So we can say this algorithm runs in the sides of linearly in the sides of the matrix. The same amount of time is required to compute the all set maximal matches within the reference panel. On the other end, computing the set maximal exact matches with an external haplotype is faster because it's just linear on the sides of the external haplotype. So it's big O of W. We can ignore the number of matches because they are. We can approximate this complexity bound to big O of W. But we cannot just focus on time. We also need to take into account space bounds. And this is the key point of the PPWT, because each type of query that consider only the reference panel, so for example, the first 2 problems, can just run while we are building the index. So we need to just store the information for a single column and output also the matches. So we have a very light data structure in terms of memory usage. On the other hand, if we want to compare an external haplotype, in this case, we need to store a lot of additional arrays that takes a lot of memory. We are talking about something that is linear in the size of the matrix, but in reality, this is linear, okay, but a lot of bytes. In fact, if you do some math, we can obtain 13 HW bytes. And if you remember the numbers that I told you before regarding the chromosome 1 panel from the 1000 Genome Project, we obtain something that is like 400 gigabytes of RAM. So it's not something that everyone can provide. Just some other consideration. The first one is already done because it was about storing just the information for 1 column per time. The second one is really interesting because Richard Darwin also developed an efficient algorithm to perform external queries that just merge the queries inside the reference panel and perform the set maximal exact matches finding within this new panel. This algorithm is not well described because it's not described in the paper. I honestly don't know why. But it's really hard to extend to solve other kind of problems. So this is the reason of the development of the MoPWT, and we will see how later. We can also solve other kind of problems. For example, set maximal exact matches with a coverage, because I mean, in the example seen before, we have some SMEM with just 1 line in the reference panel. Is this enough to provide some meaningful result? I'm a computer science, not a biologist, so I like to provide also some algorithm that put more strong results. So provide a coverage of a minimum k lines in the reference panel. And here we can see an example of k equal 3, and obviously all the matches with just 1 line disappear, and we have smaller matches in terms of column, but obviously bigger in terms of rows. Then we have another very interesting problem that is called the maximal blocks problem finding problem. And in this case, block is just like a set maximal exact match, but with some less restrictions. In fact, we can output a block every time we are losing a row. So let's look at an example because probably it's easier. If you look, for example, to the yellow rectangle, this is a block, but this is not a set maximal exact match because if we are computing the SMEM, we can extend the 0 line and the 2 line because we have one in column 5 on the other end and obtain, for example, this set of SMEMs. But if you are looking for blocks, we can just output also this internal match. Blocks should be really interesting to understand similarities inside of our reference panel, and also they are used to build, for example, Pangenome graph. We will see something later. Last problem that can be solved using the PPWT. This is the minimal position substring cover, and this is a problem I worked on during the last year. In this case, we want to cover an external query using a set of substrings that are completely covered in our reference panel. So each position of the pattern must be covered, and obviously we are interested in minimizing the size of this set of substrings. This is an example. Again, we have a strong correlation between this problem and the set maximal exact matches finding problem. We have, for example, a coverage that is exactly the first SMEM in the left. Then we are not interested in overlaps, so we can just skip the first 3 columns of this match and just focus on the last 5, and we continue until scanning the entire pattern. We have some variants of this problem in literature. We want, for example, the leftmost MPSC that consider the MPSC set that start as least as every other possible MPSC, or rightmost it's the opposite, so ends at least as late as any other. Or again, we can impose a sort of coverage requiring to have at least k rows in our panel that provide the coverage. So this is an overview of the main problems, but the development on the PPWT just doesn't stop with Richard Darpin. We have a lot of variants, a lot of extension proposed in literature in the last more or less 10 years. As introduced before, we can have the multi-allelic PPWT, so we have not focused on binary alphabet, but an arbitrary ones. Then we have the so-called dynamic PPWT that does not rely on arrays for storing the prefix and divergence arrays, but they become linked list in order to enable the insertion of new haplotype. And this can be very useful in a real-world scenario. Then we have some improvements in terms of computational time. For example, in this case, solve the L-long matches. And then we have something that is more related to compression of the index of PPWT, because data are becoming larger every day. If we think about UK Biobank data, we have panels with hundreds of thousands of rows and millions of columns. So we need to compress more also this space-efficient data structure. And also we can focus on computing in parallel, because if you remember the description of the PWT, we need to proceed column by column, but there are some projects related to how we can build, for example, starting from different point in the panel and then trying to correct every time it's not the correct index. Now a sort of sponsor for our laboratory, because this is the wild PWT developed by another PhD student on our lab. And in this case, the project is focused on finding maximal blocks in panel with errors or with missing data that are represented here in the example with a star. And the star is a sort of wildcard. It can be anything. So as you can imagine, this problem is exponential in time. And using the PWT, it remains exponential because in the worst case, it remains exponential. But it can easily run also on real-world panels with an error rate of 1, 2, or 5%. And yes, this algorithm should be used also to eventually correct the reference panel. So, just an overview of my main project. This is the MoPWT. If you are used to the last, well, not last, because the R index PWT is developed, I mean, I think 10 years ago, but okay. We tried to use this run to index the PWT, and we developed, in collaboration with all the guys you see in the first point of this list, a sort of R index for the PWT. So we just wanted to perform queries not more in linear time of the, in space, not more in linear space, but in sublinear space. So focusing on the number of runs. And the first problem we solved was the set maximal exact matches with an external query. And we wanted to outperform these 13 HW bytes. That was the trade-off, strong trade-off on using the Darpin's algorithm. This data structure is strongly based on such data structure, so it's really light, but really fast to query. And we also extended the use to solve the K-set maximal matches and the MPSC problem. I won't go into details because we don't have enough time, but just think about that each of these components that are the MoPWT are stored in a space that is proportional to the number of runs. I will skip this example because it's really too hard to describe. But let's just see the results. These blue dots represent the PWT developed by Darwin. And as you can see, for the chromosome 1 of the 1000 Genome Project data, well, chromosome 1, but also chromosome 2 that is bigger, we are talking about 400 gigabytes of RAM for performing the set maximal exact matches. On the other hand, MoPWT, that is the orange one, just require more or less 5-6 gigabytes of memory. Without any, yes, we are a little slower, but not so much. And also the index file is very light in memory if we consider that the BCR file is the binary input. We have 2 gigabytes of index compared for 0.5 gigabytes of index of input file, so we are okay. And we can also scale on UK biobank data. These are the results for the chromosome 20. And as you can see, these panels are really sparse, and more the panel is sparse and more runs and less runs it has. So this is the best scenario for us, but we are lighter than our index is lighter than the input size. So it's really, really an interesting result. And to conclude this talk, I want you to present some projects that uses the PWT to solve real-world problem. First of all, Pangeblocks, because it's developed in our lab and it will be explained in the next talk. That uses the PWT to compute the set maximal blocks, starting from multiple sequence alignment, and then uses these blocks to build a Pangenome graph. Then we have Wilms2 and Impute5 that are already described by Simone Rubinacci in the first talk of today. Then we have pSmoother that is used to correct errors in order to provide better panels for downstream analysis, but you can also perform IBD detection, again, as told by Simone Rubinacci in the first talk. And then nowadays I'm focusing on this problem, that is finding haplotype pairs in the reference panel that can resolve an input genotype. So we have also heterozygote sites in the genotype and want to find the best pairs of haplotype that resolve this input. So this is all for today. Probably I have been too fast, but I think it's okay. We have time for questions if you have something. And thank you so much for your attention.
Thanks, Davide. We're waiting to see if there are some questions from the audience. Otherwise, my first question is, while the standard Pearls-Wheeler transform is quite well known and is very widespread in bioinformatics, the positional variant is still not as widely used. Do you have any idea or any opinion of why it's the case?
Well, probably we have many problems that are related to linear reference. So we are looking for alignments, we are looking for maximal matches finding problem on a single sequence file. So a single FASTA, a single sequence. And obviously we have many problems on this kind of input instead of haplotype panels. And on the other hand, we are obtaining many more data just in the last few years. And the PWT was developed just 10 years ago. So probably we need some time to make this data structure also suitable for biologists and less theoreticians, there are people to use it. I don't know, honestly. This is my suspect.
Thank you very much. And then another question is, how do you think that those techniques can be used to index Pangenome graphs?
Well, Pangenome graph, this is probably a simpler representation of a Pangenome graph because we are skipping the common parts of, I mean, if you think about a Pangenome graph, we have, for example, a node that is shared between, for example, just a simple example, all the haplotypes, and this kind of information is completely missed in our haplotype panel. While on the other hand, if we have a bubble, we are just storing more or less the same information. But I don't know, probably a sort of mix between storing a reference and the PWT of a panel can allow some similar query, but probably the graph is the best solution in this in common tasks like alignments or exact matching.
Okay, Thank you very much. There are no more questions from the audience, so we thank you again for giving this talk.
Thanks.
And we are going to resume the session in slightly less than half an hour. See you then.
Okay, hello everybody. Okay, great. Gianluca, okay.
Okay, you ready?
Yep.
Simone, okay.
Let me start sharing.
So if you can present the first slide, the title slide, please. So
Should work, right?
Yep, great. So we are going to conclude the workshop with the last presentation, which is given by Simone Cicolella. Simone is one of the organizers of the workshop and is currently a postdoc in our university. He's an expert of combinatorial methods for pangenomics, and so we are sure that we are going to conclude the workshop on a very high note. So please, Simone, start your presentation.
Thank you very much, Gianluca. Thank you all for attending this workshop. This is the last one. We are close to lunchtime, so I'll be quick. And my talk would be on modeling and combinatorial approaches for pangenomics. Okay. What does it mean? I will continue on the same note that Davide left us on, which is more a computer science approach to this biological problem. And I would like to show you 3 different methods that we developed in our lab, with a focus mostly on how do we model biological program in a more computer science way, and how can we solve them algorithmically. So I will not go into deep learning or machine learning topics. This will be just purely algorithmic and combinatorial. However, my goal is not to be too technical. I don't want to bore you with the technical details. I just want to share with you some idea and key concepts that we can use, hopefully. So pangenomic construction is the first problem I would like to show you. I will not go into the details of what a pangenome is. We already seen it described multiple times in this workshop. But what I'm referring to is, just to be clear, it's a graph pangenome, okay? And typically there are different ways to do so. One of the first ways that we use to build pangenomes, and some tools still use this method, is to use a reference plus a VCF file. So basically we would have, in this case, a reference represented by this line, and then we have different variations, okay? And for example, in this case, this red one would be considered as deletion, okay? And what we end up is that we want to encode these genomic variants represented in our VCF file as different nodes in the graph. So we try to keep our graph as linear as possible, but we had different nodes and different links when it is necessary. This is one of the easiest methods to do such a construction, because it relies on data that we pretty much already have. We already have reference, we have multiple VCF for different populations. It's good, but it has some issue. Why? Well, because the main goal of the pangenome is to try to avoid what we call the reference bias. And if we rely on the reference NAVCF to construct a pangenome, in some sense, we are still relying a lot on the reference, and we still may have this so-called reference bias. Therefore, different methods have been proposed. One, which is the one that we will focus the most, is the one based on the multiple sequence alignment. So what we do is that we take a bunch of sequence and we do a multiple sequence alignment. So we have different methods to do so. We typically either do a pairwise mapping, and then we map all the pairwise all together and obtain we can call this as a matrix, right? We have this matrix where we have different organisms as rows, and we have the different genomic composition on the columns. This is technically better than what we have before, because at least we try to minimize reference bias since we don't have a reference right now, but we are comparing all the organisms between each other. The main drawback of this is that multiple sequence alignment is a costly operation. It requires a lot of time and a lot of resources, especially as the length of the genome increases and as the number of sequences that we want to align increases. And similarly to what we did before, we are able to construct a pangenome from this multiple sequence alignment in many different ways. Every tool has its own way to implement this, but the idea, again, here is to keep the common parts all together and then make adjustment when needed. There are other ways to do pangenome construction other than doing proper multiple sequence alignment. We can do pairwise mapping and augment at each comparison the graph. We can also do some incremental construction, which is not dissimilar to what we've seen in the talk yesterday, where we have a new organism that we want to, a new individual that we want to add to our current pangenome, and then we adjust the pangenome based on what we see. This is one of the tools that we proposed. It's called PangeBlocks. This has been published last year in BMC Bioinformatics. And here we relies on two different concepts. So we are going to use multiple sequence alignment, and we are going to use an ILP to solve this problem. Thanks to Davide, to his previous talk, we now know what are these maximal blocks. And in fact, we used a similar approach in this work where given a multiple sequence alignment, so we're not as before with a matrix of 0 or 1. Here we have different characters, and we want to compute these maximal blocks. So as explained by Davide, we want to have blocks that cover the more position as possible in both columns and rows. And the idea is that we want to cover the entire multiple sequence alignment with these maximal blocks. Now, technically, maximal blocks cannot cover all of our input. Therefore, we might have to not really use maximal blocks, but use smaller blocks to cover more. And here there are trade-offs that we can have, and we'll see them shortly. So what is the ILP modeling? So ILP, as you all know, is integer linear programming. So what we want to do is that we want to devise a mathematical formulation to solve this problem. And in particular, with any ILP model, we were going to have some decision variable and some constraints. So the basic of this is that the decision variable are binary variables in this case, where we want to see, we have variable for each block, okay? And the decision here is whether we want to use this block or not. And we have some constraints, which is, for example, each cell must be covered by only one block. Therefore, with this, we can construct a model such that each of this cell of the matrix will be covered by one block. Of course, this alone is not enough, right? Because the easiest solution would be to have a block for each of these cells, which is not good. Therefore, we also have some objective function. And I think this is the beauty of this work, alongside the actual ILP formulation, is that basically once we have this variable, we can define whichever cost function that we want. So if we have a look at this slide, C would be W and C would be the cost, sorry, not W, omega, and C would be the cost of this function. And this can be defined as we want. right? So for example, in our paper, we have a bunch of those. I'm going to show you 4, which is the number of nodes. So for example, in this case, we want to minimize how many nodes we have in a graph. Or we can minimize the length, the total length of the graph. We can also prioritize, in some cases, shorter nodes, or either way, we can minimize nodes that are too short, or we can minimize nodes that use few sequences. Okay, so this is 4 of the objective function costs that we define in this paper. And while they tend to have some similarity between them, all of them can be used to optimize a different part of the graph, such that we will result in different topology. Slightly different topology that still explain the same input. The beauty of this is that, well, first of all, it's easy to add new objective function. So if we have some specific cases for which we would like to have a specific function to optimize, we can do so just by changing our objective function cost, while the other entire modeling still continue to work. This is quite nice, and it is nice that this can be fine-tuned for each specific case that we want to study. This is nice, but still it is an ILP. So even though I really like ILPs, I think they are a beautiful modeling approach, they have a main drawback, which is computational cost is very expensive, both in terms of memory usage and time. Okay, this is not always the case, but in average, they tend to be quite expensive. Therefore, this is still a work in progress. We have made some approaches, but none of them, we haven't found the correct method yet. But the idea would be to devise an heuristic method to solve the same problem. And this, of course, would mean that we lose the guarantee of optimality that we retain with an ILP, but with the main advantages of gaining a lot of performance to solve the same problem, right? Okay, so suppose now that we solved the Pangenome construction and we want to use it. The first approach that comes to mind, the first idea that comes to mind when we want to use a Pangenome is to do, for example, a sequence alignment. Typically, we all know how to do sequence-to-sequence alignment. That's what we usually do when we want to analyze and to map reads. In this case, the problem is a bit more difficult, right? Because we have to do a sequence-to-graph alignment. So suppose we have this string and input. We will go step by step, and you will see that it's quite easy, right? Because we can start with T, we can take T, then we can take ACG, which is the next node, and we can continue this mapping as long as the sequence we want to map can be found in the graph. Of course, if we have insertion, deletion of error, we can modify something. What is something? Well, usually in most of the cases, what we actually modify is not the graph, right? But we want to modify the read. So we would obtain some different cigar, which is what we usually do when we do sequence-to-sequence alignment, right? So in this case, we do not get exact coordinate position as we normally do, but we will get this sequence of nodes in the graph that would be the mapping of our read. Now, what happens when we have a variation graph? Well, what is a variation graph? It's something that we add on top of this structure. So, for example, here I've added some colors to this graph, and this graph represents now what we call a variation graph, where we basically have different walks or paths in this graph, and they represent something. Okay, typically they represent haplotypes. but they can represent other type of information that we want to encode in the graph. So in this case, this is what we would have. So we have for each of the, let's say in this case, they are haplotype for each of these walks in the graph. We have different color and different nodes. So what happens when we want to now sequence, so to map a sequence to this graph? Well, if we have a sequence like the one we had before that perfectly matches any of the haplotype, we have no issue at all. This is all on the green haplotype and we're good to go. What happens when we have something that does not map correctly? Now we have different way to solve this problem, right? What we can do is that we can follow the paths, okay? And in that case, we will prioritize the paths of the graph. So for example, we want to map following one of the haplotypes. Here we have different ways of doing so. For example, here we have a deletion or a substitution or an insertion, depending on which path we choose. And of course, we would have a scoring function for these, and we would choose the best one for this. This is nice because it makes the problem easier. It's not that different from doing sequence-to-sequence alignment. We just have to do it multiple times following all the paths in the graph. However, we are not really fully utilizing the capability of a variation graph. Another way we can do it is that we ignore the paths, right? We do not consider them, and whatever comes out based on how close the sequence are to the graph. So we do like we did before, and we ignore the paths. That is still easy problem, but now this will require some post-processing because we have to decide what we do with these paths. We can have multiple paths, and then we have to decide which one we want. Or another approach is that we, on top of the original sequence to graph alignment, we can model recombination. So in this case, we would call a recombination moving from one path to another one in the graph. And that's what we did in this paper. RecGraph is a recombinational aware alignment of sequence to variation graphs. So the goal of this is that we want to map following the paths as much as we can, but we model moving from one path to another, and we have a cost associated with that, depending on the goal of what you want to have, right? So we can customize the weights of these operations. Very interesting in this paper, and if my memory serves correct, there is no other paper, no other method that allows to do this, which is this sort of displacement where we can move from one path to another at any point in the graph. There are methods that allow to move to one path to another, but only in nodes where the two paths collide. But with this method inside a bubble, for example, we can move from one path to another. Of course, this adds a cost to our sequence, also cost that we can fine-tune to our applications. I will not go into the detail of this, but if you want, the paper is out to read. This is dynamic programming approach, and we want to maximize a given scoring function. All these ways can be fine-tuned if you want to minimize substitution, insertion, deletion, recombination, or different displacement values. And in this work, we also tried this on bacteria. And in particular, what we try to do with bacteria is that we try to assess also with phylogenetic analysis whether this sort of recombination made sense or not. And in most of the cases, they do, and we actually find recombination to different organisms. These are simulated results in this case, but We simulated them with a specific recombination in a specific point, and we were able to actually reconstruct the same recombination point, which is good. Now, this work has a limitation. Currently, it's limited to only one recombination, and this is done optimally. And again, optimality requires a lot of resources. So what we are doing right now is to develop an A*-based heuristic to do this mapping. And also we want to allow multiple recombination such that it would be less computationally intensive. This work is not published yet, but we have a preprint that you can have a look at. So the last problem that I want to show you is the problem of alternative splicing. So alternative splicing is that this phenomenon that we see in nature where when we basically express from DNA, RNA, and then we get the protein, this translation is not always the same, right? We have, given the same DNA, we can have the, based on the condition of the replication or the environment, the same portion of genetic material can produce different mRNA and therefore different protein. And the way we usually solve this in classical approaches is that we have alongside our reference a database of all these different exons and how they combine with themselves to produce proteins. However, this is still reference-based, and all the databases are reference-based. But let's have a look at what we see usually. So suppose we have a reference, we have it annotated with this part of the reference are the different exons, okay? What we do is that we get a bunch of reads. These are spliced reads, spliced aligned reads. So we have a bunch of them. What we do is that we try to separate them in cluster and see what they actually express. Okay, so in this case, the first one would be 1, 2, 4, the second 1, 3, 4, and the last one would be 1, 2, 3, and 4. This is all well and good. And we can classify these different events based on what happens. So for example, if in the annotation we have this pattern, so we have exon 1, exon 2, and exon 3, we see the reads that skip over exon 2. We call this an exon skipping. Sorry. If we have the 2 exons, but we also see a retained part in between those, so we would call this an intron retention. So where we basically, our RNA also express the part in between those exons. We can also have points in which, for example, one exon extends over what is in the annotation. And in this case, we would call this an alternative splice site, in this case on the 5'. And similar, we can have the symmetric on the other side and have an alternative 3' splice. So how do we do this on a pangenome? This is quite new. This paper is, I think it's out. If not, it will be quite soon on PLOS computational biology. And as far as we know, this is the first method to actually do a proper modeling of this event on a pangenome. So what we do is that we want to augment the pangenome with this type of information. So let's start with a reference, just to give you an idea. So we have this reference, we have the variation in the reference, and then we have different annotation for the transcripts. First of all, we also get a pangenome. We also need a pangenome. What we want to do is that we want to up the information regarding the transcripts on the pangenome itself. And therefore, we have developed this technique that basically try to transpose the linear information on a pangenome, and we can add on top of the already present information, we can have additional information regarding these splicing events. We can also annotate this splice pangenome by including into it the actual information on the transcript and the haplotype, so that we have all the different possible combination inside of our pangenome. Therefore, once we get a read and we align this read with a spliced alignment, we can follow what happens in the graph and actually deduce the type of the event that we see. So the main modeling contribution of this work is that we define a framework when we can basically call these different events. So if we start with annotated events, this is a different representation of the exon skipping that we saw before, but is actually saying the same thing, but instead of a linear reference, this is done on a pangenome. Unfortunately, it seems like I cannot use a pointer, but basically we have the blue squares are the exons, the small white squares, unlabeled ones, are the introns, and then we have these edges that represent the different splicing events on top of the edges that represent the actual genomic pangenome. So we have exon skipping, we have the 5' splicing site that you see is expanding on the right. Similarly, the 3' is expanding on the left. And intro-retention, here we see that we have a bigger transcript that comprises all of the nodes. This is for annotated events, and we basically get this information from a database, but it also works for novel events, so events that we don't really have annotated and we discover when aligning the reads. I will not go into the details of this modeling because it's too complicated to see this in a slide, especially without a pointer. We tried this method, we tested on both simulated and real data, and we compared it against reference-based methods that are the only methods that we have. We don't really have any other method to do so on the pangenome. And we actually showed that using a pangenome is first of all feasible, and we obtain very good accuracy, both in precision, recall, and all the measures that we tried. So not only is possible to do it on a pangenome, but we can do it with high confidentiality of our results, and especially when we compare with the with the other methods, also on true validated events in the lab. This is all good. The main drawback for now is that these methods rely on having a splice alignment to a sequence graph, or to a pangenome graph, let's say. And currently, there is only one method that allows to do so, which is VG-MPMAP. And that method requires a lot of resources. So, but our tool is independent of the type, is agnostic on the type of splice reads that we obtain. So we hope that when more and more people start working on this type of alignment, better and faster alignment tool will come up, and we will still be able to get even more out of this work and even more out of the pangenome. So now this is the most important part of the talk. So the boring stuff is over. Advertisement time. What are we going to do right now? So These are the next events that we have. The first one is CPM 2025. This is going to be in Milan, in Italy, in June 17 to 19 this year. You're all welcome to participate, and I think the submission deadline is past, not sure. But if you got accepted or if you want to join us in this beautiful city, you can come and we would be happy to host you. Now, more specific for the 4Genome project, which is the sponsor of this workshop, we will have a PhD school plus an hackathon this year in July. This date should be correct, meaning that there is the time frame. It's not strictly defined yet, but if it's not this exact date, it will be around that date. So would be not that far to that. So we will have 4 days PhD school, and the topic would be Introduction to Bioinformatics Services in Pangenome, Cancer Genomics, Introduction to Modern Genome Sequencing Technology, Data Analysis and Software Tools in Genomic Structural Variation, the latest trends, methods, and experimental validation of this data. And we will have a very nice, I think, part that would be especially useful for early postdocs and PhD students, whereas in a more generic way, not specifically for research, but what are the research challenges, how to do this is really hard, entrepreneurship. And this is going to be trained by Amble, which is one of the partner of this project. And as I'm pretty sure you all know, they're quite good and quite strong in what they do. So it would be very interesting. And following this, we will have a 3-day hackathons. This will be on genomics, proteomics, and bioinformatics. For now, we have some confirmed speakers for the school, which are Piotr Prince, Eric Garrison, and Enza Colonna, all of 3 from University of Tennessee. And you may know them. You most likely know at least Eric Garrison, which is one of the main proponents of the VG toolkit for variation graph and for Pangenome. And we're going to have Tobias Rausch, which is a bioinformatician from Amble. Lastly, thank you for attending this workshop. The live streaming recordings are already available on the website. You have the link. We sent to you in both in email when you registered. It's on the website, and we sent on the metrics. And for now, these are just the live streaming recordings. Edited video will be available soon. We hope next week. And lastly, thank you all for attending to this talk and for attending to the whole workshop in general. I would like to thank 4Genome2, which is the sponsor of this workshop. And these are the partners of the project, which is Comenius University, Amble University, the University of Milano Bicocca. And thank you for the European Union to funding this project. So if you have any question, I'm happy to take them.
Thanks, Simone. It was very clear presentation of the current status. One quick question is, what of the several problems that you have described, which one do you think will be the most challenging in the next few years?
I think both the most challenging and the most important would be the sequence to graph alignment. The reason why I say this is that we have a lot of methods that build Pangenome, and I mean, pretty much they all give us some solution which is good in practice. But as we've seen with reference genomes, we know that the main goal, the main challenge is actually to get an alignment of the sequence, because this is what all the downstream analysis rely on. And we saw a lot of methods to do sequence to graph alignment, but none of them still solves all the issue. We don't really have a clear understanding on what we want exactly from the graph and what we want this alignment to be. And this is why we see a lot of alignment tools. And each of them has its pros and cons. It is specifically devised for a particular problem. And either we want to have more of those to solve specific problems, or we may have an established one that can encompass all of these issues. But right now we don't really have a de facto standard, and I think that would be the most important and the most challenging because it's hard to do so.
Okay, thank you very much. It was very, very clear, very relevant. I think that we can conclude this first annual workshop. Thanks everyone for participating, and I'm sure you will disseminate these videos once they are fully published on YouTube. Goodbye, everyone.
Thank you all. Goodbye. Finito!